again. The session here is Socio-Ecological and Environmental Systems. And we will start with the first presentation with Stephan Peter, who will be talking about or presenting a paper entitled Climate Change and Necessary Actions.
Okay, thank you. It's a great pleasure for me to be here today and to meet so many interesting people and to be able to give this talk. Indeed, it's not a paper, it's just a talk about, which is about climate change and necessary responses. And we've already heard a lot about, but I'm sure I will add some new perspectives. Okay, climate change is related to water scarcity. So we know that currently more than 2 billion people in the world lack clean drinking water, at least for some periods in the year. And not us, humans lack clean water, also the plants. So, and this directly affects the food security. And yeah, it's clear that without any action, it will not be better, get better, the situation in the world. Yeah, why? Because as it was mentioned before in some of the talks, humanity uses more than 2 Earths, the resources of more than 2 Earths each year. So we exploit the planet, and the climate shows us in response that it's not good what we do. And I think it just began what we experience now with increasing floods, with increasing earthquakes, drought, heat waves, and so on. Yeah, and on the other hand, there is a lot of funding on different levels, regional, national, international. So there is a lot of money for projects. And now the question is, how should we use this money to combat the climate change? Yeah, and what we propose is large-scale greening, which means desert greening, afforestation, and supporting agriculture with the newest irrigation technologies. And yeah, here I put some statements which apply. So we would apply principles of permaculture, so combining different plants which are proven to work well together, to grow well together. And what's the aim? The aim is to establish a stable vegetation cover within 1 or 2 growing seasons. Why is this necessary? Because then the moisture in the earth is increased, and this will affect the microclimate in a positive way. For example, it will lead to a cooling effect of the air. And by this, we hope to get valuable information for entrepreneurs and other stakeholders, and to provide scalable solutions to combat increasing drought and moisture deficits. And we want to perform a pilot project, and a local university or Department of Agriculture should do the scientific documentation to collect this data about growth of plants and how environmental parameters change. This is what we propose, and our approach includes a trained intuition. And there is the NATA training since 45 years in Germany and internationally, which provides this training of intuition. And it's a complementary resource for gaining information and idea, which will be useful to perform such complex desert greening projects. They are very complex, and as we know, the complexity we can solve some problems with machines and AI, but human intuition is able to provide more than this. It is able to provide really new information and ideas, which no one had before. And I will show you on the next slide some examples for this, real-world examples, how we applied intuition to get solutions which really work and help to start these large-scale greening projects. So also I will mention the documentation which was done about natal training, which was neuroscientific measurements of the brain during you train your intuition with this training program. Okay, so 4 examples I will show. First, the olive plantation in Tuscany in Italy, and then natal energy. It's an invention which was made by applying natal approach. And then I will talk shortly about mental influence on microorganisms, and to activate vegetation, in this case, to activate forest flora. And then I will talk about the neuroscientific approach, proof. Okay, so more than 30 years ago, in the beginning of the 90s, the Swiss architect Rolando Santini, he bought a property near Florence, and he wanted to create an olive plantation. And as usual, he asked Mr. Philip Latan for advice. He did it usually for personal and professional matters, trusting Mr. Latan's intuitive insights. Mr. Philip Latan is the co-founder of the Natal Institute, which, as I said, exists since 45 years in Germany. And he now provided some information for Mr. Santini. One information is that is about the variety of olive trees. So he recommended to use a variety of olive trees which was deemed unsuitable by experts. And the second thing, Mr. Latan explained that the 480 olive trees had to be planted not row in row from left to right, but he pointed the positions for planting one after the other, and they were not easily row in row. And as I said, Santini trusted these advices, and he stayed with the farmers who did the planting, because he knew if he would have let them alone, they would have done what they want. They would have planted as always, row by row. So these are 2 examples of the information Mr. Latan provided by his intuitive approach. And we can see 30 years later, we have this olive plantation, and we have some results. And only 3 out of these 480 planted trees died out. This is 0.6%. And typically about 20% of the trees die. So he had an exceptionally high rate of success in terms of vital olive trees. And also the quality of these oil Santini is producing has exceptional quality. And this is proven since many years. We see on the next slide a selection of some of the awards Santini is winning since many years for his oils. As a conclusion, I want to state that, of course, these intuitively gained news represent a challenge for corresponding theories, but they are also an opportunity to develop these theories and models further. This is important, and for the example of these olive trees planted, not row in row. Maybe we can compare it to acupuncture which is successfully applied in medicine, especially in Chinese medicine. And yeah, this is one example of how intuition gave really useful information to perform a project in agriculture in this case. So now we come to the second example. It's natal energy. And natal energy is developed a container, which you see here on this slide, which uses solar energy to produce electricity, water for drinking or for irrigation, and it also produces warmth, so heat or cold, for air conditioning, for example. And how does it produce water, for example, sustainably? It can draw water from air. And the second opportunity, which is more effective, is the desalination of seawater. Of course, this makes only sense if you are near to an ocean. And yeah, these multi-purpose supply containers, they were developed by using this intuitive approach. And we did a publication about the performance in different climate zones, which is there in the lower right, I mentioned it. And we saw wherever you have sun, they work really well, and this concerns most regions in the earth where most people live, especially Morocco. Oh, one slide is missing here. Yeah? Okay. Yeah, it's a nice slide. This map was not good. Really? Maybe because of Western Sahara and Morocco Sahara. Oh, we did not check this. Sorry to all Moroccans. Yeah, we developed with my students at University of Applied Science in Jena, we developed an online simulation tool for computing the amount of energy and water we can produce each day a year. For this, we use NADA weather data. We have a contract with NADA for this, and we have access to the data of the last 30 years and also to weather of today, the weather and precast. So what we can do with this, if you plan to create an agricultural plot and you want to use water from desalination, we can compute how much solar panels you need and how many of these containers, if you want to put the technology into containers, you would need to provide an irrigation which secures the water supply for the plants. Yeah, and usually there's a world map and you can click and you can have the information. And yeah, this you get on the second click, you get all these diagrams. It's a screenshot from December 2024. Yeah, on the upper right, you see the energy production for each month, the daily energy production. And it was for Rabat here, and I think it was around, yeah, an average around 70 kilowatt hours per day. And on the lower left, you see the water production by desalination, and on the lower right, you see the water production by atmospheric water harvesting. There are different unities, so atmospheric water harvesting is a lot of less efficient, but if you have no other source, it is useful. So the third point is not yet published in a scientific publication, but it was, yeah, it regards some experiments performed in the 1990s by Dr. Kleespies in his laboratories, and they were observing Legionella. And on the right, you see 3 images. First shows the population in the beginning of the experiment, the second after 28 hours, and the third after 50 hours. And after 50 hours, there were no longer virulent Legionella there. And the only thing which was done is that Mr. Latan focused on these microorganisms and treated them mentally. And it would be very interesting to study what's going on in the brain during this process. A second experiment was about two forest plots. They were selected by a state-certified expert, an agricultural engineer. He didn't know what the experiment was about. He had only the task to select two very similar forest plots. And after 1 year, he was asked to do a protocol, to do some measurements and a protocol of what was happening. So he analyzed the soil, and he analyzed the plants and the insects and so on. And I cannot show the whole protocol here. It would exceed this presentation, but there is a very remarkable sentence in the end, translated to English. It is, all in all, the treated site can be described as more vital than the untreated zero area. He found there were more microorganisms in the soil, the plants had more roots, and there were more insects on the treated area. Again, just by mental activation of the forest plot. Yeah, and now for the scientific proof. So the NATA training was established in 1980, and from the beginning, Mr. Latan and his wife, they asked neuroscientists to do neurological measurements, for example, EEG measurements of the brain. And yeah, I only extracted some images here of these studies. So this is before the training. You see left and right hemisphere. So the horizontal axis, there are different frequencies which are active in the hemispheres, and the vertical axis, it's time. And you see left and right hemisphere, the image looks quite different. They are not synchronized. So when the NATA training starts, you see that the synchronization begins between left and right hemisphere. And now I skip some images. This is in the full NATA training. After maybe half an hour, you have these images. So you have synchronization of the brain in all frequencies, and it's also proven that new synapses can be formed. It was shown the first time in the 80s, and mainstream scientists only confirmed this possibility in 1997. And we would like to do an update of these measurements with new technology. So after 30 years, the technology is much advanced, so it would be very interesting to do at any university an update of these measurements, because there are many questions still not answered about this. And I would like to now show a video of 5 minutes, an interview of this scientist, Dr. Haffelder, who did this analysis, this neuroscientific analysis. And this was part of a TV documentation of the second largest German public TV channel. It's called ZDF. And the then head of the Department of Science and Art, Karl Schnelting, he was doing this training, he was participating, he was completing this whole NATA program, and then he decided to produce this documentation. And on the next slide, I would like to run, if it's possible, this interview with Dr. Haffelder. It's a bit outdated technology, but it's very interesting what he says. Can we start it? Okay.
Was waren das jetzt für Experimente, die in Weißenstein mit Dr. Philipp Ebrard gemacht wurden?
Bei Dr. Philipp Ebrard war eine Besonderheit, er hat versucht, über Gedanken, über eine bestimmte Einstellung, Materie zu verändern. Er hat versucht, Kristalle zu verändern, und er hat versucht, Pilzkulturen über seine Gedanken schneller wachsen zu lassen. Dazu haben wir ihn eben an das EEG angeschlossen und konnten feststellen, dass er ganz bestimmte Frequenzmuster aufgebaut hat, die er gepulst hat. Das heißt also, er war nicht nur in einem Zustand, sondern er hat quasi so eine Art Höckerbildung erreicht, hat immer wieder die Pulse rübergeschickt an diese Objekte. Und wie wir nachher auch feststellen konnten, hat sich etwas verändert. Also für mich ist Dr. Evra ein Phänomen, er kann ja zweierlei machen. Er kann auf der einen Seite, wir sagen dazu, Kontakt zu anderen Ebenen aufnehmen. Das zeigt sich in dieser langsamen Sinuswelle, die sich bei ihm im Beta-Bereich ergibt, die eine Schwingungsfrequenz von 1 bis 3 Hertz hat pro Stunde. Also nicht Hertz, sondern 1 bis 3 Schwingungen pro Stunde. Das ist eine sehr langsame, die sogar teilweise langsamer wie Erdbebenwellen, die aber vollgepackt ist mit Informationen. Bisher konnten wir diese Welle noch nicht entschlüsseln. Wir können sie sichtbar machen inzwischen, aber noch nicht entschlüsseln. Das kann Dr. Evrat erstmal auslösen und gleichzeitig eben, dass er jetzt auf Gegenstände Einfluss nehmen kann. Auf Gegenstände, auf Pflanzen und so weiter. Da gibt es bestimmte Gehirnbereiche, die er aktiviert, also sehr gezielt aktiviert. Und durch diese Aktivierung scheint es, hat er Zugang zu diesen Pflanzensystemen oder zu diesen Mineraliensystemen.
Könnte man denn auch annehmen, dass er sozusagen Zugang zu anderen Informationsebenen hat?
Wenn er diese langsame Sinuswelle aktiviert hat, hat er diesen Zugang. Also das ist etwas, das etwas sehr Seltenes ist. Wie gesagt, wir können es noch nicht entschlüsseln. Wobei natürlich bei Dr. Evrat etwas sehr Außergewöhnliches auftrat. Er bringt es fertig, also zum Beispiel seine Schriften zu machen. Beim normalen Medium, das channelt oder in andere Ebenen kommt, muss ich mich darauf verlassen, dass das, was es sagt, einigermaßen stimmt. Denn bei Dr. Evrat habe ich die Möglichkeit, ganz genau herauszufinden, dass das erste Zeichen entspricht dieser Gehirnschwingung, das zweite Zeichen dieser. Ich kann es also ganz genau zuordnen und dadurch ist mir wissenschaftlich natürlich etwas gelungen, dass ich Ähnlichkeiten vergleichen kann mit ähnlichen Gehirnschwingungen. Und von daher eventuell einen Schlüssel finde, diese langsame Sinuswelle zu entschlüsseln. Also ist auch für mich eine Besonderheit und ich bin sehr dankbar, dass ich ihn da messen konnte, denn das führt uns ein Stück weiter in dieser Forschung, die wir normalerweise eben doch sehr subjektiv führen mussten seither. Und durch Dr. Evrat wird es uns möglich, es sehr objektiv über diese Schriftsymbole zu entschlüsseln.
Ist Ihnen denn schon mal was Ähnliches begegnet bei anderen Leuten?
Diese Sinuswelle bei sehr guten Medien, ja. Aber natürlich, wie gesagt, eben sehr subjektiv.
Dann denkt man ja bei dem Wort Medium oft an Trance, an einen Zustand des Unbewusstseins. Ist das bei ihm auch der Fall?
Ja, das ist wiederum ein Unterschied. Dr. Evrat kann sich in Trance versetzen, bei ihm tritt diese Welle auf, selbst wenn er ganz normal mit mir spricht. Also ich kann mich normal mit ihm unterhalten und gleichzeitig kann er diese langsame Sinuswelle erzeugen. Das heißt also, er hat während des Sprechens, des Normalseins, Kontakt zu anderen Ebenen.
Also das Wachbewusstsein ist aktiv in jeder Phase?
Voll aktiv, ja. Er kann dabei Rechenaufgabe lösen, er kann sich mit mir über irgendetwas unterhalten und trotzdem ist bei ihm diese Sinuswelle vorhanden. Das habe ich in dieser Form noch bei niemandem entdeckt.
Es ist ja auch charakteristisch auf den Bildern, dass er, wenn er sich jetzt...
Okay, may I have 3 minutes for concluding remarks? Okay, merci. So, I'm talking about intuition. This is nothing new. Everyone who invents something or has a very good idea, there is intuition in work. And in our publication, Intuitive Innovation, which I showed here several times, unfortunately not here on this screen, I quote a lot of scientists from Einstein, Marie Curie, Newton, Poincaré, Pascal, and so on, who said which role intuition plays in their work. And they say it's the most important thing. It's more important than logic and ratio. Ratio comes after intuition. Intuition is the first thing. Without intuition, you don't have the initial idea. And NATA is a way to train intuition and to improve it. Yeah, here I want to show quickly on some of the colleagues I work with. There's Philip Latan on the upper left. He's a co-founder of NATA, and on the right there is Maria Frisol. She trains NATA in France and in Arab countries. And on the lower right there is Professor Bashar Ibrahim. He's the president of Gulf University of Science and Technology, and I do the publications with him. And on the lower left there's Georg. He is process engineer, and he was much involved in developing these containers. And yeah, finally, I want to summarize which could be the next steps for NATA-related research at the university. So there's first a pilot project in desert greening and agriculture. Second, updating these neuroscientific measurements from the 1990s. This would be very interesting. Many things are not decoded. Third, to apply NATA training at the university for professors, for scientists, researchers, staff, students, to do a conference. What do we have? 2 times the number 3. Yeah, we develop NATA health systems. It's another topic. The next point is NATA manuscript. You saw these exotic scriptures. We have 11,000 pages of these, and we still don't know what it is exactly. We are working with students and AI, but it's still not deciphered. And yeah, last is research about mental influence on microorganisms, and there would be much more. If you are interested in collaboration, just contact us. And yeah, shukran. Thank you very much for your attention.
I guess thank you, Mr. Peter. We will be moving to the next presentation. If you have any more questions, I'm sure you'll meet and you can be discussing them later. The next presentation is Professor Luis Comolli presenting a paper entitled Emergence in Designed Agroecosystems, Collective Dynamics in a Perennial Multi-Species Agroforestry Network.
From which can I? Hello, yes. So my name is Luis. I'm going to be presenting our agroforestry system from where this picture was taken. So what we see here is a farming road in the center and these bush-like plants on both sides. Those are Ilex paraguayensis, in South America known as mate, a typical infusion, a sort of green tea in South America. And intercalated with them, this line of trees. So moving on, the whole purpose of this experimental system is to assess the correlation between different species of trees and the behavior, the performance of these ilex plants. So first, I want to start highlighting the establishment of the system, highlight the main results, move on to an agent-based model built from experimental data collected here from the system. So it's a realistic agent-based model. To finally highlight an approach using networks and max entropy to close the knowledge gap between simulations and experiment. So first, the context of the study. I highlight here in bright green the province of Misiones between Uruguay, Paraguay, and Brazil. And there to the right, with the red pin, a UNESCO World Heritage Site known as the Jesuit ruins of San Ignacio, and the yellow pin, the site of this experimental system. Do notice the darker green color of the province of Misiones, this region, and it's one of the few known advantages of the dismal economic performance of Argentina. The rest is light green because agriculture took over with annual grains while we were left with a lot of subtropical jungles. Okay, so these are the main goals: to prevent soil degradation, to prevent irreversible fertility loss, if possible to restore degraded lands, to restore part of the biodiversity native to the region, and all of these avoiding landscape simplification and restoring biodiversity. The system was started before the current situation with the acceleration of climate change, so today part of the objectives include carbon sequestration, adaptation, and mitigation to climate change. Okay, highlights of the system implementation. In 2010, we decided to start the experiment with 10 hectares and 9 species of trees selected for the hypothetical synergies that we, from experience, from intuition, decided were good candidates. Those are highlighted with an asterisk in this list. The list covers more species, 20, because after the success of the first 10 hectares, we expanded the whole experiment to 200 hectares today. And this is the original design of the system. These 10 hectares were divided in 4 sub-blocks, B1 to B4, and the 9 species of trees plus 1 area without trees acting as a control were randomized in these 4 blocks so that then we can acquire systematically data with scientific relevance from them. These result in about 1 hectare per species and per control. And this is the design. So the cross indicates plants of mate, the red diamond, the trees to be evaluated, and the mate plants were installed at 1.5 meters between each other in 5 lines, also separated by 1.5 meters. Let's see if, yes. These 5 lines of mate were separated between each other by roads of 3 meters in width, and in the center, the line of trees also separated by 1.5 meters. The result of this design is 3,700 plants of mate per hectare and 740 trees per hectare. So we are dealing here with about 4,500 plants per hectare. So for the 10 hectares, it's close to 50,000 plants. I'll highlight some of the main results. This is a view of the system after a few years. Top A and B, we see then the harvest, the target harvest plant, the mate, with intercalated trees. And the point is to note that while these trees have been pruned of the lower branches, the pruning of the branches results in the incorporation of organic matter. They are left on the ground. 2 of the 3 trees have been cut down in order to manage the regrowth of the new trees, to later on cut the trees that outgrow to a big size. And bottom left, a picture from another lot that was established earlier to experiment with this. We can see the ground covered with docile herbs, trunks and branches lying on the ground, and the crowns of the trees producing partial shade. Bottom right, a view from another perspective to indicate that we also incorporate annual grains to have the soil always covered, ryegrass in this case. And here I establish a comparison of the performance of the system during a climate shock. in 2022, we had the worst climate shock on historical records, with 5 months of drought in a region where drains are monthly, and 42.5 degrees at the end of the drought period. So we can see at the top that there is a stress, the yellow color, but the canopy is green, evergreen, and the harvest plants in the context of the trees are very resilient. The whole system is resilient. At the bottom is the performance of a monoculture. The trial lots left as a control experiment. And we see brown branches, and branches die back, considerable stress and death of plants. So this was not this aspect, adaptation, mitigation, of course, reduction of the local temperature, conservation of humidity, was not really one of the original purposes, but these results show that these objectives are very important. And this is the result of the cumulative harvest of ilex for each of the associated species and the control, integrated over 10 hectares. So we have the result of the harvest in 1,000 kilograms per hectare. So number one, there is a very strong production performance across all the species and the control. But we also see that for some species, the total is higher than the control and higher than the median. For instance, Araucaria, Lapacho, Petirivio, Loro Negro, and also the exotic species Kiri from Japan, and Tona, an Australian cedar. For some species, aboriginal species, Watambu and Angico, the harvest is lower, but just 12% lower. So there is a strong performance of the composite system. And this is the result of the harvest per associated tree species per year, indicating the high variability. So we measure the whole system during the establishment, the initial evolution of the system. And notice that at the beginning, Tona, Australian cedar, is the highest in 2015 associated harvest, while at the end, native species take over. More numerical results. We also measure the count of insect populations, in particular this insect, a xylid called Gyropsila, within the system as in comparison with the monoculture. And systematically, several measurements, different years and different lots, we get approximately half the insect count within the system as with respect to the control without trees. This is because we have birds, and as a consequence, we don't even use fertilizers, insecticides, sorry. And this is perhaps one of the most important numerical results. We systematically acquire soil samples and do soil tests for complete profile. I extracted just the total organic matter and phosphorus, and we see at the end of this measurement period an increase of about 10% in total organic matter and a doubling of the available phosphorus concentration. So then the system really does achieve this goal of having stall soil degradation. Clearly, we can prevent fertility loss. And so that's also an objective achieved. Now I just want to highlight aspects, views of the system to have a better grasp of what we are talking about. So at the top, I'm used to having a pointer, so that's what leaves me walking here. At the top, we see the control experiment to the right, without trees, adjacent to the sublot with trees. Top right, the the system during winter harvest period, partial shade, soil covered with docile herbs. At the bottom here, a bird, a worker pruning trees. All these pruned branches go to the soil. And to the right, during spring, you can appreciate this in this area, the diversity of species, flowering or not. So I mentioned the recovery of biodiversity. We also installed a camera trap system for 40 days, recording 24 hours a day. And we verified the recovery of small mammals within the agroforestry system, which they are absent. 5 minutes? 3 minutes? Oops, wait. So we see the recovery of small mammals in the system. These are more views, just to highlight that biodiversity also improves work environment. At the top, 2 pictures during the climate shock with dry grass, the nursery producing seedlings for the system, and at the bottom, routine working experience during the year. So the system provides a climate moderation, a much better work environment, also incorporating skills and knowledge at the worker base. So now I move on to simulations. So I had 3 minutes to 15, but I have 20, right? I cut questions and that's it. Okay, so we'll have shorter questions. Okay, now I move to simulations. During the establishment of the system, we also measure allometric parameters. And here I show a table of the diameter of breast height and height of the trees. And to the right, plots showing the great variability and the change in rate during the growth of the trees. With these parameters and other measurements, we can build a netlogo model. This is a field of 2010 by 2010 pixels, where I embed 20 by 20, a grid of 20 by 20 ilex plants. That's 400 mate plants, and the 9 species of trees plus the control experiment. These are simulations running for 10 years. We have at the top left the intact trees, different colors. To the right, we see already human interventions with pruning and cutting, reducing the density of the trees. The soil starts to change. The color indicates the soil quality. Bottom, then the appearance of structural heterogeneity and an increase in the quality of the soil indicated by the color. Notice that the ilex plants, which are the tiny ones, are bigger within the areas of trees. So we have this mosaic of growth, regrowth, and soil changes during the evolution. Here I show the dynamical evolution of some of the main state variables for the system. The state of the soil initially decays, as the experimental case, to then increase, and the harvest reduces its increments during the deterioration of the soil to then recover. Bottom left, the total biomass evolution. I noticed that the steep increase coincides also with the recovery of the soil, increase in the harvest, and as a result, the ratio needed of external fertilizers to harvest decreases, which allows a better economical result. This is the harvest per tree species in the simulation, per average per tree, and in darker green, I indicate the cases where there is a match with the experimental case, and in lighter green, where we don't agree with the experiment. These 2 species in particular, Angico and Guatambu, were lower than the median, and in the simulations, they are higher. That means that the parameters incorporated in the agent-based model are missing something. So then, to address this, I move to networks. Each plant has 4 nearest neighbors and 8 second nearest neighbors. And we can assign weights to these links to produce a graph that evolves in time with changing weights on the links, positive synergies, negative competition. So bottom right, after 2 years, we see the initial deterioration of the soil. And to understand what's happening, how the micro-interactions give rise to the emergence of patterns, this is an area of 50 by 50 pixels, because there are 8,000 links in total. So we see a mosaic pattern of synergy, positive interactions top, then gives rise in red negative interactions, calling for interventions, the redistribution of interactions, bottom left, and the resurgence of competition in red. Closing now, this is an evolution, graphs of the evolution of community structure, just showing a slice of 2,000 of the 8,000 possible links. Notice the, again, distribution of synergies that gives rise to clear patterns of antagonism and synergies, calling for interventions, and a appearance, again, of antagonistic interaction. So the point here is the community structure is functionally determined. We can also analyze measurements of the structural complexity, and in all cases, entropy of the node strength, harvest entropy, the divergence or correlation, we see a sharp transitory period at the beginning that then later on stabilizes, reaching a sort of plateau. So eventually, as with the species reach a stable set of interactions. And this is the last slide. Then to close the knowledge gap, looking again at what is missing in the model, we know from the data that each tree species results in different free phosphorus concentration. We know that the quality of the shade is different because some leaves are fractal. And we also know that below the ground there are fungi. So we introduce 3 potentials to see if we can go from the priors, the weights of the simulation, with these new potentials and iteratively optimizing the Lagrange multipliers for these measured data, re-estimate the harvest per species. And the answer is that these potentials do correct the results to match the experiment. And so, as conclusions, the system does hold soil degradation, avoids landscape simplification, reincorporates biodiversity, provides microclimate regulation, adaptation, mitigation, expands the knowledge base for workers, and the design of the system, because of the systematic way it was planned, is easily scalable. We went from 10 to 200, and it can be transformed to different regions, different people, different regions with different cultures and different modes of doing things. These are the people that work throughout the years in the system. At the top, the 2 engineers following weekly the system, and at the bottom, the National Institute of Agricultural Technology. And these are the publications.
Thank you. Thank you, Professor. Now we'll move to the next presentation by Shinnosuke Yoshikawa, who will be presenting on behalf of his team a paper entitled Socio-Ecological Evaluation of Synecoculture Implementation in Sub-Saharan Africa.
Okay. So good evening, everyone. My name is Shinnosuke Yoshikawa, and I'm from Synecoculture Association and Kyoto University in Japan. Today, I will talk about our studies. The title is Socio-Ecological Evaluation of Synecoculture Implementation in Sub-Saharan Africa. And this is joint work with Funabashi and Ota from Sony Computer Science Laboratories in Japan, and Langshi and Tindano from African NGO. So let's start with the background. In African countries, about 90% of farms are smallholder farmers. These small farmers produce about 80% of its country's food in some cases. So they are very important for local food security. However, smallholder farmers in Sub-Saharan Africa face serious problems, like climate change is hitting this region harder than anywhere else. And many farmers are caught in a poverty trap. They are too poor to invest in better farming methods. This is a creator cycle. Poverty leads to poor farmings, which leads to more poverty. So this is a serious global issue that we must address. Okay, so what is Synecoculture? Synecoculture is a new way of farming. So it grows many different plant species together at the same time in the same field. It combines introduced plant vegetation with naturally growing vegetation intentionally and uses ecosystems' ability to self-organize and support plant growth. This is make it possible to farm without tilling, without fertilizer, and without pesticides as a result. So instead of optimizing conditions for each plant species, Synecoculture aims for an ecological optimum as the complex systems. As a result, the greater the diversity of plant species, the higher the productivity becomes. This method was also featured in the World Economic Forum white paper in 2025. We studied Synecoculture in Africa in 2015. The first site was Burkina Faso in the Sahel region. We used 150 local plant species. The result was remarkable. Local farmers resolved desertification in just 1 year. The land also recovered its forest ecosystem. After this success, we expanded our work. We now support more than 50 fields across Sub-Saharan Africa. We also established the name of CAFS, the African Center for Training and Research in Synecoculture. CAFS now leads the training and expansion of Synecoculture. In this study, we worked with 3 major programs. The first is the ABL DMPLC program. This was funded by the Embassy of Japan. The focus was infrastructure support for local sites and human security. The second is the ATSA program. This is supported by Sony Computer Science Laboratories. CAFs provide training in certain African countries. This project's leader is Andre Tindano, co-author of this paper. The third is UNEP CTCN program in Northern Cameroon. CTCN is the Climate Technology Center and Network. The budget was $175,000 US. We established 12 Synecoculture fields. The program focused on women's economic empowerment and climate resilience. This project leader is Luce Langshi, also co-author of this paper. Let's share the screenshot of official introduction movies. Some of our study sites were in politically unstable regions, such as Northern Cameroon and Burkina Faso. In these places, complex approaches like Synecoculture may work well. Synecoculture uses local natural processes and local educational school. This can help people produce food even under difficult social and environmental contexts. These 3 programs together created our 27 study sites. Our survey covered many different contexts. Based on questionnaires and interviews conducted on site or online, we collected data on 8 positive factors, things that help Synecoculture succeed, like harvest counts, like a consumer's number, like a yield rate. We also collected 12 negative factors, barriers that prevent success, like social barrier, economic barriers, and ecological barriers degree, and 11 other relevant factors. This survey gave us a comprehensive picture of each farm. So we measured success using 8 variables called Y1 to Y8. Y1 is the farmer's own evaluation, like a subjectivity. Y2 measures necessary conditions, like species densities and harvest frequency. Y3 measures sufficient conditions, like productivity and training quality. Y4 combines Y2 and Y3. Y5 and Y6 compares the result to our benchmark farm in Mahadaga, Burkina Faso. Y7 measures yield per cost, how much crop you get per money spent. Y8 is the benefit cost ratio. This is like a profit measure. Y8 above 1 means the farmers earns more than it spends. These 8 indicators give us a complete picture of Synecoculture performance. For analysis, we used 2 statistical models. The first is a generalized linear model, we call it GLM. The second is a logit model, we call it LOM. LOM separates factors into 2 factors, positive factors and negative factors. And GLM add other factors. One important feature, all coefficients in our model must be positive, because this ensures we can clearly identify what helps and what matters, what hurts. We use 4 different optimization methods to make the result robust. Now, let's look at the results. The model showed strong performance. The GLM achieved R squared above 0.76 of mean model's result. The logit model achieved R squared value above 0.70 of mean model results. Both results were statistically significant, with p-value below 0.01. The figure at the bottom left shows the model fitting for each response variable Y1 to Y8. The x-axis shows the predictions data, and the y-axis shows the original data. We also validated the models using farms in Japan. Sites that were not used its training model. The Japanese sites also fit the model well. This suggests that our findings may be applicable beyond Africa, like Japan. And a key analysis methods we use is called shadow price. And shadow price shows how much each factor affects the outcomes of Synecoculture. For example, we can measure how much Synecoculture changes when consumers or educators increasing by 1 unit and compare their contributions. It is calculated from the coefficient of each models and their partial derivatives. A large positive shadow price means that the factor provides strong effects. Okay, the most important findings, the biggest negative factors are economic barriers, especially like labor cost and seed cost. These hurt performance the most. On the positive side, the biggest positive factor is plant species diversity. Specifically, the number of useful plant species introduced. This was positive across 4 indicators. This is a very clear and consistent message, like introduce more plant species and outcomes improve. We also ran our model without economic barriers, because in some contexts, economic barriers are hard to change. So we want to see what else matters. The results were revealing. 1, even without many problems, ecological factors still drive outcomes. 2, plant species diversity remains the strongest positive factors. 3, training quality also became more visible as an important driver. 4, this tells us even if even if we can't fix poverty immediately, we can still improve outcome by increasing biodiversity and improving training locally. These are actionable interventions. So let me share some interesting results from specific sites. Our benchmark farm is in Mahataga, Burkina Faso. Its benefit-cost ratio Y8 is 2.1. Now look at the new site's performance. Sotobua in Togo achieved Y8 of 3.0, higher than the benchmark. Pamtanga in Burkina Faso achieved Y8 of 2.3, also higher. Gondala in Ethiopia has the best productivity scores Y2 and Y4. Athena in Chad and Antananarivo in Madagascar also showed positive returns. This result shows that Synecoculture can succeed in many different African contexts. Our result revealed 2 important discussion points. First, the poverty trap is confirmed. Economic barriers dominance as the biggest obstacles. This is consistent with its existing literature on Sub-Saharan Africa. Second, we found a perception gap. Farmers gave themselves higher scores in self-evaluation Y1, but object measures Y2 to Y6 showed lower scores. This means farmers may not fully recognize some barriers, especially social barriers like knowledge gaps and training needs. This perception gap is important for designing effective support programs. Based on our findings, we recommend 3 priorities. First, increasing crop species diversity. This is the most effective and controllable intervention. It does not require large money and investments. Farmers and trainers can do this directly. And second, address economic barriers through external fundings. Labor costs and seed costs remain the biggest obstacles. International programs can help provide subsidies or support. Third, improving training quality and knowledge transfer. The perception gap shows that better training is needed. Together, these 3 priorities create a pathway to resilience. Synecoculture offers a variable, low-input solution for climate-vulnerable Africa. Yeah, let's try. Yeah. To conclude, let me summarize our key messages. This is the first large-scale statistical evaluation of Synecoculture in Africa. We analyzed 27 fields across 14 countries. Our GLM and LOM models showed strong explanatory powers. Economic barriers are the biggest challenge. The poverty trap is real. But plant species diversity is a powerful and consistent positive factor. Some new sites, like Sotobua, already outperform our benchmark. This shows Synecoculture can succeed across many African contexts. We believe Synecoculture is a promising solution for food security and climate resilience. Our next step is to expand the network and continue monitoring. Yes, thank you very much for your attention.
Thank you. We will move now to the next presentation by Shuntaro Aotake. He will be presenting a paper entitled Measuring Plants Biodiversity Across Scales, the Alpha-Beta Scale Diversity Model with Image-Based AI Estimation.
Okay, perfect. I'm so very sorry to wait, keep you waiting, and thank you again. Good evening again. My name is Shuntaro Aotake from Sony Computer Science Lab, and thank you very much for joining. And today I will present our work titled Measuring Plant Biodiversity Across Scales, the Alpha-Beta Scale Diversity Model with Image-Based AI Estimation. Okay, so first of all, for the background, the ecosystems that exist on the Earth are structures built upon the complex network of the interactions among all the plants, animals, insects, decomposers, and the microorganisms that comprise them at various scales. For example, when herbaceous plants grow, they create shade, and the insects that eat those plants form communities. When trees grow, the birds gather to eat their fruit or perch on their branches to feed on the insects below. Since plants' roots secrete unique substances, specific microorganisms like fungi colonize them, and within diverse vegetation, these organisms also form their own communities. There are also the concept known as the augmented ecosystem and the Synecoculture. An augmented ecosystem redefines the human activities, technology, and social systems as part of an ecosystem, and refers to the state in which human involvement augment biodiversity beyond its natural state, achieving a stimulus optimization of productivity, sustainability, and diversity. Synecoculture is an agricultural ecosystem design method based on high-density, multi-species intercropping, and serves as a concrete implementation of the augmented ecosystem. It is a method for establishing agriculture by forming an augmented ecosystem and harvesting from it. Since these methods result in a state of vegetation diversity that exceeds that of natural vegetation, there's a need for indicators that can analyze these states in detail. So, ecosystems are very difficult to analyze properly due to their complexity, but plant diversity serves as one effective measure of them, as simple indicators of how many plant species are present and the degree of their uniformity. There are several traditional indices that are widely used, such as the Shannon Index, Simpson's Index, and the Hales Index, and so on. What's interesting about this plant diversity is that precisely because everything is connected, it holds the potential to inter everything else from its information. Instead, the key limitation of the traditional indices is their dependency on species identification. This leads to high cost, low frequency, and poor scalability. It is also difficult to integrate with remote sensing technologies. Thus, a species-free and scale-aware approach is needed. In particular, as someone who worked in the field, which is myself, I feel that the task of species identification requires an extremely high level of expertise that the general public, lacking such knowledge, could never hope to perform. Moreover, the work of wading through vegetation in natural ecosystem is extremely arduous. Consequently, there is a challenge in that vegetation analysis often cannot be scaled up or progressed effectively. That is precisely why this study proposes indicators and methods for analyzing ecosystems that do not require specialized expertise for observation and that utilize technologies such as sensors and AI to make the process even simpler, more efficient, and scalable. Before we go on, I prepared a very short quiz. This is one of the Synecoculture field, and this red square represents the 30 square meters. How many plant species do you think are growing in this area? With existing methods, it is necessary to accurately identify the scientific names of these plant species and record details such as ground cover percentage. If even a single species remains identified, calculation may be fail or accuracy may decline. I'm sure that is a demanding process. And the answer is there are 70 species. It's very difficult to make precisions based on the image, but even if we were on the site, it would be very difficult to observe this value also. It requires specialized knowledge of plants and eating. Even with the deep learning-based recognition, it can be done right away. So we introduced the biodiversity index that does not require taxonomic identification, explicitly model scale dependence, and can be operationally estimated from the aerial RGB images using AI. So I will explain my research by dividing into the following 3 sections. The first one is the proposal of the new metric. The second one is to the evaluation and the validation of the new metric. And for the last, the validation of automation using AI. So our key idea is to view biodiversity as a function of spatial scale. We build on a well-known species-area relationship in ecology, which describes how the richness of species increases with the area. This function can be approximated by the straight line when both of the x and the y-axis are converted to the logarithmic scale. Therefore, on this approximated line, let the alpha be the logarithm of the number of plant species when the x-axis value is 1, which represents a 10 square meter, and let beta be the slope of this line. In this case, alpha indicates local plant diversity, and beta indicates spatial heterogeneity. We propose this index as the alpha-beta scale diversity index. Incidentally, this figure shows that the blue points and the blue regression lines shows the area represent data collected from the Synecoculture field with extremely high plant biodiversity. The orange points represent the Synecoculture field with the moderate plant diversity, and the red point represents the natural vegetation. And the green one represents the data from the conventional farming method. If you look at the plotted points and the regression lines, you can clearly see the differences. So this formulation has 3 key advantages. First, it does not require species identification. And second, it explicitly captures scale-dependent biodiversity structure. And third, it can be directly integrated with image-based AI methods. Okay, as for the chapter 2, for the validation, we use a data set from 3 types of ecosystems. Conventional farming field, natural vegetation, and the Synecoculture field. Excuse me. The total samples of the fields are 97, and each of the data are observed by the 2 square meter area and 1 3 square meter. The data are obtained on the several countries, including Japan, Ghana, Cameroon, and Senegal. And this is the result of plotting each field samples on the alpha-beta plane. The Synecoculture fields are plotted by green, natural vegetation is orange, and the conventional farming are blue. Then I use the support vector machine to segment each field. The result predicted as Synecoculture is yellow area, natural vegetation, blue, and the conventional farming as purple. We achieved the classification accuracy of the 93% of Synecoculture and 82% for natural vegetation and 43% for the conventional farming. One reason for the low classification accuracy for the conventional farming is that many of the observed fields were practicing the conventional farming by the family. And in such field, many types of plants is cultivated within a small area, making it difficult to distinguish them from the low diversity natural vegetation. However, commercial fields practicing conventional farming on the large scale are reliably classified on the position in the purple area in the lower left. Next, the reason why the identification rate for the Synecoculture is high with the natural vegetation at the ground around about 80% is that first, the Synecoculture is identified as long as it possesses a certain level of diversity. It results in a high identification rate. And second, the boundary between the Synecoculture and the natural vegetation is ambiguous. And there are cases where the relationship reversed between Synecoculture and the low Synecoculture with the low implementation level and the natural vegetation with a high diversity. However, if we view this figure from the perspective of the degree of achievement of the augmented ecosystem, we can treat each of the benchmark for comparing that degree of achievement. We also compared our method with the classical biodiversity index. The results show the strong consistency with the Simpson, Shannon, and the Hale indices. The left figure is the alpha scale index, and following to the right, the beta scale index, Simpson, Shannon, and the Hale index. Synecoculture is shown in the green, natural vegetation in blue, and conventional farming is orange. In simple terms, the Simpson index measures the influence of dominant species. The lower the value, the more diverse the vegetation. The Shannon index measures overall diversity, including evenness, and higher value are better. The Hale index simply indicates the number of plant species. In terms of the ecology interpretation, Synecoculture fields showed extremely high biodiversity. For example, on the Shannon index, it reached high value that is close to the theoretical maximum. This suggests that the Synecoculture vegetation achieved high species richness, high evenness, and weak dominant structures. The previous results were based on the verification using the data collected manually. But what if the area we want to analyze were much larger? In the quiz earlier, the area was just 30 square meters. But when it comes to the analyzing ecosystem, we might easily end up analyzing the 10,000 square meters. And ultimately, we want to analyze all the land on the Earth. So, to automate the estimation process, since simply attempting to estimate the data is difficult with current technology, we decomposed the proposed indicators and the estimation targets and designed them as a process. First, we estimate the number of plant species per 1 square meter from the overview RGB image. And here, we use a deep learning image recognition model for the estimation. Next, based on the number of the plant species per square meter obtained spatially, we use the multi-scale regression model to estimate the number of plant species when the target area is increased. From the plotted area of the estimated number of the species per area, we estimate the approximate linear line in the logarithmic space and calculate the alpha and beta. The result shows that the first, we achieved a mean square error of 0.68 for species count prediction by using the deep learning image recognition model, which is the efficient net. Then, by predicting using the multi-scale aggregation, alpha can be estimated with the 88% accuracy and the beta with the 76% accuracy. These results demonstrate the practical feasibility. In conclusion, we proposed a new biodiversity indicator that does not require species identification and captures scale-dependent structure. By integrating this model with AI, we enable to scalable and automated biodiversity monitoring. As for the future work, first, we will expand the dataset and validate and enhance the AI. And next, extend the method to satellite-based observations for real-time ecosystem management. And third, integrate with the data from the other attributes, which are 3D data, multispectral images, and the thermographic image data, and also IoT sensor data will be also used. And by conducting an integrated analysis of these multi-data, we should not only be able to improve accuracy, but also estimate other aspects of the ecosystem simultaneously and from multiple perspectives. That is for my presentation. Thank you very much for attention.